Dear students, today let us discuss about the physiology of respiration. Respiration in mammals or pulmonary respiration involves three phases. Firstly, breathing or ventilation. Secondly, transportation of gases by blood. And thirdly, internal or cellular respiration. Regulation or control of breathing mechanism involves both neural and hormonal control. Let us see the transport of respiratory gases. Respiratory gases, which are the gases transported in a living organism or the living system, are oxygen and carbon dioxide. Oxygen is required for various physiological activities, whereas carbon dioxide is released after metabolism from tissues. The exchange of gases occur between the lungs, alveoli, and blood capillaries, that is ventilation of respiration, and at tissue level between cells and blood capillaries. Let us start with the transportation of oxygen. Oxygen is very important gas essential for cellular respiration. It is transported from the respiratory organs, that is lungs, to the body tissue by an ion containing respiratory pigment called hemoglobin present in the RBCs. This pigment is called as oxygen carrier. When one, two, or three oxygen molecules are bound, it is said to be partially saturated. In humans, 97% of oxygen diffuses from alveoli epithelium and the endothelium of blood capillaries into the blood. Only 1.5 to 3% of oxygen is dissolved into the plasma. At the level of tissue, oxygen level is low and carbon dioxide level is high in blood carrying oxyhemoglobin. Readily dissociates to oxygen and hemoglobin, which is also called as the Bohr's effect. Now uh, let us discuss about oxygen dissociation curve. The oxygen dissociation curve is a graphical representation when we plot partial pressure of oxygen on x-axis and percentage saturation of hemoglobin on y-axis. There are various factors affecting oxygen dissociation curve. They are the carbon dioxide, pH, temperature, organophosphate compounds, and body size. Now let us discuss one by one. The effect of carbon dioxide on oxygen dissociation curve. The rate at which the hemoglobin reversibly binds or releases oxygen is regulated by several factors, and one such factor is carbon dioxide. At tissue level, because of metabolism, there is increased production of carbon dioxide, and as a result, there is increase in the partial pressure of carbon dioxide, that is, which comes up to 45 mmHg. During when there will be rapid dissociation, the uploading of oxyhemoglobin releasing oxygen to the tissue. Rapid dissociation of oxyhemoglobin to release oxygen in the presence of carbon dioxide is called Bohr's effect. That is, increased concentration of carbon dioxide causes more oxygen to be given by blood, given up by oxyhemoglobin at a given oxygen pressure. When the partial pressure of carbon dioxide to 80 mmHg, the oxygen dissociation curve shifts to the right and shifts to the left when the partial pressure drops low as 20 mmHg as shown in the graph. Hence, Carbon dioxide lowers the affinity of hemoglobin to oxygen. Now, let us discuss about the Haldane effect. Deoxygenated hemoglobin combines more rapidly with carbon dioxide than oxygenated hemoglobin. That is, when the partial pressure of carbon dioxide readily combines with hemoglobin in the blood as bound carbaminohemoglobin or hemoglobinic acid, the bicarbonates in plasma. This phenomenon is called as Haldane effect. In the diagram, you can see that the Boer's effect and the Haldane effect have mirror images. Now, let us discuss about the root effect. When the acid strength is very high in the blood due to the maximum concentration of carbon dioxide, the dissociation curve not only shifts to the right but it also becomes completely flat. You can see that in the diagram, which is bluish in color. In this condition, at any high oxygen tension, there is no chance that hemoglobin reaches saturation level. This profound effect of pH on oxygen dissociation curve is called as root effect. Thank you, students.